Hello and welcome back. We are rounding up the end of the topics that we need to look at for this semester and saving the best for last. We are beginning our look at the gonadal systems, sometimes more commonly referred to as the reproductive systems. You will see me refer to the gonadal systems and then also refer to separately something that I'm going to refer to as reproductive biology. I split it into those two very defined categories, and we'll talk a little bit more about that as we get into those lectures. One of the other things that I like to include within this is a introduction, if you would, into how the gonadal systems developed. And so this lecture and the lecture after this is going to look at something called embryogenesis, the development and formation of an embryo, which really encompasses about the first 8 to 10 weeks of development. Once you reach week 10, then you begin to transition into fetal development. And so everything that you want to know about the human body is really implanted and cemented within those first 8 to 10 weeks of development. After that, what essentially happens is that developing embryo turns into or transitions into fetal development. And it's kind of like taking one of those sponge dinosaurs and you pop it in water, and then, like, over the next 3 to 4 days, it keeps getting bigger and bigger and bigger and bigger. That's what's happening during fetal development. But prior to that, all of your organ systems, all of your hormonal regulations that's going to drive development is all happening during those first 8 to 10 weeks. And so we're going to spend this lecture on understanding what is supposed to happen. And then the next lecture, we're going to kind of look at what happens when you've got variation in the patterns of embryogenesis, specifically around gonadal development. It's a fascinating story. And if you want to understand why the body does what it does, we need to be able to understand the processes that are happening during this really critical period of development known as embryogenesis. And so let's go ahead and jump into this. Again, I'm going to divide this into two videos. This first video is just going to look at what happens during development, what happens during the gonadal developmental process. And then the second video, we're going to look at variations in this process. And so with that said, let's go ahead and jump into this. And we're going to start with just kind of understanding the basics of what happens here. And so sex determination, or gonadal determination, is really regulated by a series of genes. And there are two that are the most influential. The SRY gene, the sex determining region of the Y, that's what SARI stands for, that gene usually gets the most attention. And it is typically viewed as being kind of the driving gene, or the gene that is really regulating the entire process. That is a little bit of a misrepresentation of what happens. It is true if there is a Y chromosome that is present. So the SRY gene, hence its name, sex determining region of the Y, that SRY gene is present on the Y chromosome. And it is responsible for the development of male gonadal anatomy. In other words, it pushes the development of the testes. And once the testes are developed, then testosterone can be produced at a higher level, which is going to drive the differentiation of tissue. That's going to lead to things like the epididymis, the vas deferens, the seminal vesicles, And later on down the road, it's going to influence external anatomy development as well. And so, from the perspective of male gonadal development, yes, SRY is everything, right? And we're going to look at and talk a little bit more about this enzyme called testis determining factor, or TDF. SRY, the SARI gene, codes for the production of testis determining factor. And it's testis determining factor that is really going to, let me come in here real quick. Pen color, what color do I want? We can leave it on red. It is testis determining factor right here that is really going to then influence the development of the testes from tissue that we're going to look at here in a few minutes. Right. And so if SRY is present, then testis determining factor will be produced under common developmental patterns, and the embryo will develop male gonadal anatomy. If the SRY gene is not present, or if the SRY gene is not, let me kind of come in here real quick here, right? All right, so if it is not present, or if not producing TDF, or if TDF is, we'll say, malfunctioning, right? If it's not functioning the way that it's supposed to, then if any of those three scenarios happen, then the embryo will develop female gonadal anatomy, right? Now, it's not as simple as that, because on the X chromosome, we have something that is referred to as the DAX1 gene, the dosage-sensitive sex reversal adrenal hyperplasia congenita critical region on the X chromosome, gene number 1. Right, that is what DAX1 gene stands for. Dosage sensitive sex reversal adrenal hypoplasia congenita critical region on the X chromosome, gene number 1. Right, and so that DAX1 gene is found on the X chromosome. And what happens here is XY, right? So males, genetic males, XY males, have one copy of this DAX1 gene, and one copy of DAX1 is not strong enough to shut down the SRY gene. And so no changes happen here. Nothing is going to deviate from this, right? SRY will still code for TDF. TDF will drive the differentiation of tissue that we will talk about here in a few minutes into developing the testes, and then from there the rest of the gonadal anatomy. But in genetic females, in individuals that are XX, it now has two copies of that DAX1 gene, and the concentrations of the DAX1 gene product is high enough that it's going to stimulate ovarian development, or the development of the ovaries. And here's the kicker. It stimulates the development of the ovaries from the exact same tissue that could possibly then become the testes. And so the testes and the ovaries are homologous structures. They are derived from the same embryological tissue. The difference is, in the presence of SRY, TDF is produced, and TDF is going to drive the production of the testes. In the absence of SRY, you've got overexpression of the DAX1 gene, and that takes that same exact tissue and forces the development of an ovary. Okay, now, what happens if, let's say you've got a genetic male, XY, and in that genetic male, so you've got an XY male, the Y chromosome is secreting TDF, but the X chromosome is hyper secreting DAX1. What would happen? Right? What would happen? What would happen if you've got an individual whose genetic chromosome is XXY, where now you've got, again, hypersecretion of the DAX1 product, still in the presence of the Y chromosome? What would happen? Right? What would happen? These are questions that we need to look at, and this is kind of where we're going to go in the second video, right? This is why when we look at sex development or gonadal development, it's a spectrum, because it's not as simple as to say, well, this is what your genetic sex is, or this is what your anatomical sex is. There's a lot of gray that gets mixed in here. And this is why, right? We put forward this understanding that, oh, it's just the SRY gene, and if SRY is there, TDF is there, and if TDF there, it's going to produce male gonads. And if it's not, magically female gonads develop. It's not that simple. And so we need to look at that. We need to understand that in order to understand why we see variation later on in life. Right? And so that is one of the things that we are going to look at. That's one of the things that we're going to look at. So let's jump into this process. So for the first 8 weeks of life, actually for the first 7 weeks of development, of embryological development or embryogenesis, the developing embryo is actually sexually indifferent. You'll sometimes hear that, oh, we all start out as females, right? That is inaccurate. We do not. We do not, and I'm going to be obnoxious and underline this like a gazillion times, start out as females. This is not, put a line through it, exit out, not true. We are sexually indifferent. And the reason why we are sexually indifferent is because each of those developing embryos, all of us, for the first 7 weeks of development, actually contained 2 sets of sexual ducts, of embryonic tissue that could become either male gonadal anatomy or female gonadal anatomy. We all contain that. And so we're not all females initially. We are actually sexually indifferent. We are neither anatomically male nor female. And there's a fascinating story that we're going to get into here in a few minutes about this. And just as a side note, this is kind of my area of expertise, right? Fetal sexual development, embryology as it relates to gonadal development and sexual differentiation is my bread and butter. This is where a lot of my enthusiasm, passion, and previous research interest has been. And so, yes, expect a lot of excitement during these next few videos, because there's a story to be told, and there's a story that has been grossly misrepresented and oversimplified to the point of fault that has been perpetrated beyond the walls of science. There's a lack of understanding of what's happening here. So things that we're going to go through here really is going to shed light on why we see such a greater push, on understanding sex as being a spectrum characteristic. And I'm going to talk more about that here in a few minutes. But we all start out being sexually indifferent. We all start out having these things called mesonephric ducts, or what we call Wolffian ducts. And it's these mesonephric ducts that we see here, that we're going to look at here in a few minutes, that will become male gonadal anatomy. So we're talking about things like the vas deferens, we're talking about things like the epididymis, we're talking about things like the seminal vesicles, right? And then we've got these other things, these other set of tubes that are called the paramesonephric ducts, right? Or the Mullerian ducts. And guess what? These have the potential to become, you guessed it, female gonadal anatomy. So all of us have both of these tubes. Now, we just got done covering the urinary system, right? Sorry, my excitement's coming through just a little bit. But I want you to pay attention to this. Here's an aha moment. Look at these terms. Nephric. 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 Nephron. Nephron. Urinary system. The ductwork, the tissues that will become gonadal anatomy starts out as urinary tissue. The embryo is recycling tissue. And both the mesonephric duct and the paramesonephric duct have very specific jobs prior to week 7 in the developing embryo that buys the time for the ureters and the bladder to fully develop. It needs time. We don't just conceive, we don't just fertilize an egg and then voila, boom, ta-da, we've got ourselves a little human. All of these organ systems, all of these tissues have to differentiate and respond to signaling pathways and develop over a period of time. And instead of wasting that tissue, our body has evolved a mechanism to recycle that tissue. And so this mesonephric duct serves a completely different purpose weeks 6 and 7. And will become, in week 8, potentially, the male gonadal anatomy, in the presence of an SRY gene that is effectively producing enough TDF and undersecreting the product of the DAX1 gene. And the paramesonephric duct has a completely separate function related to the urinary system and waste removal in week 6 and 7. And in the absence of TDF, or in the inhibition of TDF, if expression of the DAX1 gene is elevated enough to shut it down, that paramesonephric tissue will develop into the female gonadal anatomy. We're recycling it because by week 8, you have ureters, you have a urethra, you have a bladder. And here's what's even more mind-blowing about this, right? You actually developmentally have 3 different developmental stages of the urinary system. This mesonephric and paramesonephric ductwork that we're now talking about here, we're going to look at here in a few minutes, is actually the second iteration of your urinary system. Your first iteration of the urinary system, which doesn't develop until about week 4, week 5, going into week 6, is actually in the cervical region of your neck. And it's what we refer to as the pronephric kidney. Your original urinary system was in your neck. And then in the week 6 into week 7, These other tissues begin to develop that are known as the mesonephric and paramesonephric ducts. And then by week 8, your third kidney or your third urinary system develops. And that's the metanephric urinary system, where the kidney is fully developed, the bladder is fully developed, the ureter is fully developed, the urethra is fully developed. Or at least developed enough that it's going to complete development. Remember that differentiation within the male gonadal anatomy that we need to pay attention to. So again, this is a fascinating story of development, right? And so testes develop in week 7, week 8-ish. Right? If maybe the developing embryo is a little slow, it might go into week 9 a little bit. And that is because right around day 60, the Leydig cells, and we're going to talk about these in the next set of videos that we're going to look at, right, after we go through this. The testes have to develop first, so the SRY gene has to express the TDF to allow the testes to go ahead and develop. Leydig cells are a type of cell that are within the testes that are found between the tubular structures that are producing the sperm. Though Leydig cells, or interstitial cells, have to develop, and then they begin to secrete testosterone, and then the testosterone that is produced, as we're going to find out, will then later lead to further development of the testes, development of the epididymis, the vas deferens, all of that, right? The scrotum, all of that. And then we'll look at that. That's a whole nother fascinating story. In the absence of elevated levels of TDF, ovaries develop, but the ovaries are delayed a little bit, right? Ovaries develop week 9, week 10, because you got to wait and see. You got to wait and see if the testes are going to develop. If the testes don't develop, then the ovaries kick in. But you don't want both of them developing at the same time, because who wins? You got to give it time. And so there's a little bit of a delay that's built in to our development. This is why I say the embryonic stage of development is week 8 to week 10-ish. Right? Because by the end of week 10, the ovaries have developed, if conditions are conducive to that development. Right? If conditions are conducive to that development. So here is a picture of all of you for what you look like in and around week 6 of development. And I want to highlight or point out a couple things here, right? And so we've got these structures that are coming in right here, right? These long hot dog looking structures that are located right here. Right? This is what we call the gonadal ridge. Those two gonadal ridges are what TDF, or the byproduct of the DAX1 gene, is going to act on. In other words, it's the gonadal ridge that has the potential of becoming the testis or the ovaries. Right? That's where that's coming from. Right? Here we've got those mesonephros. That's right here, the mesonephric ducts that are right here. Right? These little ball-like structures that you see there, that's the glomerulus. That's what your glomeruli looked like in that second iteration of the urinary system. Right? And then you've got the duct. Right? That is right here. This here could be analogous to the ureters, right? Because it's going to filter out the blood within here. That filtrate's going to come down into here, and this is your developing bladder right here. Right? And we don't have a urethra yet. We have a cloaca, and we can go into that a little bit more. It's a little bit beyond the scope of this class, but we don't start out with a urethra and an anus. We have a cloacal opening, kind of an all-purpose, if you would, exit point for the body. Right? And so you can see right here, this is going to tie in to right here. Right? This is that secondary urinary system that we're talking. This is the tissue here that will become the epididymis, that will become the vas deferens and the seminal vesicles. if the gonadal ridges develop into the testes due to the presence of TDF because of the SRY gene that has not been suppressed by the byproduct of the DAX1 gene, right? That's present on that 1X chromosome. Right? This tube over here, check this out right here, this tube over here, got a second one over here. These are the paramesonephric ducts, or those Mullerian ducts. And what happens is this coelom that will eventually organize itself into the abdominal cavity is producing fluid, right? You get excess fluid that develops in there, and this fills up with fluid, and it's a exit point up here. When you reach this level up here, that excess fluid flows into this, it drains into this, comes down this tube, and look, empties into the bladder. That's right here. These here are those metanephros that are developing. These are your kidneys that are actually beginning to develop. This is the start of that third and final iteration of the urinary system. But look, they're not fully developed. Takes some time. Right? And so these structures right here, over here, those mesonephric ducts and paramesonephric ducts are doing the function of the urinary system. That paramesonephric duct, in the presence of SRY and TDF, this is going to absorb. It's going to go away, right? To allow for the formation of the male gonadal anatomy. If there is no TDF, or TDF has been suppressed and the DAX1 secretion is elevated, then guess what? The mesonephric duct disappears, the gonadal ridge forms an ovary, and you have further development of the paramesonephric duct into the oviduct, aka fallopian tube. Right? You also get the formation of the uterus and the upper 1/3 of the vagina. Right? All of that is going to form out of the paramesonephric. Right? Again, up here, you've got the epididymis that is going to develop. You've got the vas deferens that are going to develop. And you also have the seminal. Sorry, my pen is not keeping up with my speed here. Seminal vesicles that are going to develop. All right? And so you can see the presence of, or the lack thereof, that SRY concentration. And this here just kind of shows that developmental process that is happening along this side here. It is interesting to note, right? Over here you've got male gonadal anatomy that is developing. Over here you've got female gonadal anatomy that is happening. But both this is the right side, right? And this is the left side. Right? This is the right side. This is the left side. Both of these are developing independently. So you could have overexpression on the right, you could have underexpression on the right, overexpression on the left, underexpression on the left. You could have an absence of receptors to bind to TDF on the left, which means you're going to develop an ovary on the left, but normal expression on the right, which means you're going to form a testis. Right? You got all of these mix and matches that can go ahead and happen as a result of these development of the gonadal system. Right? But again, in general, what's going to happen here, if the ovaries are going to develop, that is going to be an absence of elevated levels of TDF, right? If the testes are going to develop and the associated other structures are going to develop, that means that you've got elevated adequate levels of TDF due to the presence of the SARI gene that is there, right? Or the SRY gene that is going to be present. And so how did we get here? How in the heck did we get to this point of development? Well, where is this gonadal ridge, this gonadal development occurring? Well, let's continue with the self-portraits from when you were a little teeny developing embryo. And we can see here we've got 3 layers of development. We've got this layer here. We have a second layer right there. And we have a third layer of development right here. Right? This here is what we refer to as the ectoderm. This here is what we refer to as the meso or middle derm. And then the inner layer here is the endoderm. And you can see that development, right? You can see this starts to fold in. This starts to develop a little bit more. Here's a picture of you by the end of week 2, maybe going into week 3. Right, this here is that kind of blown up. And where we are really focused in at is this area that is right here. Right, so this is posterior. This here would be the development of the umbilical region right here. This is the amnion that is going to be over here. This is what's left of the amniotic sac that is quickly disappearing over here as the heart is developing here with blood supply. This is your brain that is over here. All of these things are kind of in place. Well, this here is going to be the site of gonadal. This is where that gonadal ridge is going to be located. All right. And so let's take a look at this in a little bit closer of some detail. So what are we looking at here? Well, here, right here, we've got the gut tube that is present. And right here, and right here, we've got those gonadal ridges. We've got those gonadal ridges that are right there. And what will end up happening here, if we blow this up a little bit, cells, what we call germ cells, from the gut tube begin to migrate upward and out into the gonadal ridge. Right? So these germ cells from the gut tube begin to populate in the ridge of this gonadal tissue. And what ends up happening is we see differentiation of those germ cells into 2 distinct regions. The lighter color that you see there will develop into what we call the medulla. And the darker color that you see down here forming will develop into what we call the cortex. All right? The other terms that you will sometimes see for this is the cortex being referred to as the mesothelium, and the medulla here, that lighter color, being referred to as the meso or mesenchyme. Right. Mesothelium, this here would be an kind of a connective tissue that's acting as a epithelial tissue, and then the mesenchyme being more of a true authentic connective tissue, right, that is present. And what we end up seeing here is the start of male gonadal development, because what will end up happening here is this medulla area right here begins to press its way down into the cortex. And so we see these grooves that begin to kind of penetrate down into the cortex or into the medulla. Right? And those penetrations, those medullary tissue that's coming down into the cortex, those cords are what we refer to as gonadal cords, right? Or sometimes you'll see them referred to as testis cords. And this really begins, here's those testis or gonadal cords that are coming down in here. So this here again is the medullary tissue. This here is the cortex. That medulla is coming down into the cortex. That is right there. And what you essentially are seeing here is those medullary or those gonadal cords begin to loop up. And the formation or the bending, the twisting of those cords are what are going to eventually form the seminiferous tubules. These seminiferous tubules become the site of sperm production. This becomes the site of sperm production, right in that area. And these white specks that you see in here, those white specks that you see in there are what we call Sertoli cells. And the Sertoli cells are essentially stem cells that have the ability to be highly mitotic to form the sperm, right? And so these medullary cords begin to extend down into the cortex here. They begin to wrap themselves up into these coiled regions that we call the seminiferous tubules. And we get this, these cells, these clusters of cells called Sertoli cells that begin to populate the outer edges of these developing Sertoli cells. Right? And so these are the areas where the spermatogonium or the developing sperm are going to develop from throughout the individual's life, right? Throughout the individual's life. And then in this area that is surrounding here, within the cortex here that you see, we'll have other cells that will become the Leydig cells. And it's, remember, those Leydig cells will be, again, in here. And it's the Leydig cells that will produce testosterone, that will then act on the Sertoli cells within the seminiferous tubules to produce the newly developed sperm, eventually. And if it's female gonadal anatomy, we take that same gonadal ridge. Now, instead of being exposed to TDF, it's being exposed to byproducts of the DAX1 gene. And instead of forming those gonadal ridges that are forming, those gonadal cords that are forming, they still circular up, but instead of developing into seminiferous tubules, they develop into follicles. And so follicles are analogous to seminiferous tubules. Follicles in the ovary are analogous to seminiferous tubules within the testes. And in the center of each of these individual follicles is a germ cell that is an ovum. And so that ovum is really, in the ovary, is analogous to the Sertoli cells that are forming within the seminiferous tubules within the testes. Right? And again, here you can see the disintegration of the mesonephric duct and the continual formation here of the paramesonephric duct into, here's the little fimbriae that are going to be able to capture the ovulated egg once time is ready, and this becomes the oviduct that is here. If you come back, what you're seeing here is you're seeing the disintegration here of the paramesonephric duct here, and the further now development of this is becoming the epididymis, and this is becoming the seminiferous tubule. And all of this is forming out of the mesonephric duct that we see there. Right? And so again, all of this is happening right here along this gonadal ridge that is happening. Same tissue, right? Different products, different enzymes are essentially stimulating that development that is happening here. Right? I have just a few more little things that I want to discuss, and then we're going to end. I know this video is a little bit longer than typical, so we're going to kind of go through this here a little bit quicker. It's in the 8th week into the 9th week. If assuming the testes have developed, that we'll see an increase in the production of testosterone. And testosterone has the capability of being transformed into other byproducts. And so we take testosterone and we can create something that's called DHT, dihydrotestosterone. We can also, so DHT is if you would, a metabolite or a breakdown product of testosterone. Ironically, estradiol or estrogens are also a byproduct of testosterone breakdown. In fact, the ovary can produce elevated levels of testosterone that can then go ahead and be converted into estrogen to support follicular and ovary development, right? This is one of the things that causes polycystic ovary syndrome, is elevated secretion of testosterone within the ovaries of individuals. And so during male development, this testosterone is going to be converted into DHT, right? There's an enzyme that regulates that. And in the presence of elevated levels of DHT, the penis begins to form, the scrotum begins to form. So that external genitalia begins to form. With lower levels of T, because remember, ovaries can still produce testosterone, lower levels of testosterone, we're going to have then elevated levels or higher levels of estrogen or estradiols. And then what we end up happening is the formation of the clitoris and the labia minor and labia minora. The clitoris and the penis are analogous structures. They come from the same tissue. Right? The scrotum and the labia minor and majora come from the same tissue. And by the way, this is not something you need to be freaked out about overly. This is just showing you here. Here you've got the formation of testosterone that in the presence of 5-alpha reductase, we'll talk a little bit more about that in the next video, you've got the development of DHT. That is right here. Right? And if testosterone is exposed to aromatase, which is the other enzyme, then you've got the production of estradiol or estrogen. And so testosterone, in the presence of 5-alpha reductase, you get DHT. Testosterone, in the presence of aromatase, you're going to have the development of estradiol, right, or estrogen. And so testosterone is that precursor, if you would, to the formation of those other critically important hormones to drive the development of the external anatomy. And this is another self-portrait of what we all kind of looked like in and around week 8 of development. And so we all have this very sexually indifferent external anatomy that's going on. This here is the genital tubercle. This area right here is what we call the labioscrotal swelling. And then we've got the urethral slit with the urethral fold that is here. So this here is the urethral fold that you see right here. And in the presence of DHT, that genital tubercle develops and forms the glans penis. And the urethral folds will go ahead and essentially kind of elongate upward and work to form the shaft of the penis. And the urethra will form going up the shaft. Right? In the absence of elevated levels of DHT, then you've got the glans clitoris or the clitoris that develops. So the penis and the clitoris develop from that same structure that is called the genital tubercle. So up here, yes, you've got that genital tubercle. Then what we see here is the labioscrotal swelling, this yellow area right here, which will continue to fuse in the male and develop into the scrotum. In the female, that labioscrotal swelling is what is going to become the labia majora that you see right there. And then that urethral groove that you see here in the pink, this is what's going to become the spongy urethra in the male, as you've got extension of that glans penis developing right in here. Right? And that same structure here in the male is what's going to become the labia minora. It's going to remain unfused. that is right here. And so again, the glans penis and the shaft of that penis is going to be analogous to the glans clitoris, and that is coming from the genital tubercle. The labial scrotal swellings that you're seeing up here is going to develop into the scrotum, which is also analogous to the labia minora, that is right here. And the urethral groove that you see right here is going to become the spongy urethra, which is going to fuse itself within the shaft of the penis that you see developing there. And that analogous structure here in the female is the labia minora, which remains unfused, which remains unfused. And so you can see kind of that side by side right here. This here is the undifferentiated or indifferent, sexually indifferent structure. And again, you can see how we're reusing that tissue to form both either or, and also male genital anatomy or gonadal anatomy and female gonadal or genital anatomy. Quick note here, hypospadias. Hypospadias is a condition where you've got irregularities in the formation of the urethra. And so sometimes males are born with various openings to their urethra, right? And so the urethra is supposed to extend all the way up the shaft of the penis to the glans penis that's all the way up here. Sometimes the opening of that urethra forms a little bit lower. And that is due to underproduction of DHT. Right, now, what could cause that? Well, what could cause that is decreased levels of 5-alpha reductase. Right, so if you're not producing enough levels of 5-alpha reductase, you're not going to convert enough testosterone into DHT. And that low level of DHT is not going to be able to stimulate the production or the development of that urethra all the way down the shaft of the penis to the glans penis. And so, again, a lot of room for things to not work the way that we expect them to. Now, this here is something that can be corrected with surgery. They'd go in and they would actually create the pathway for the urethra and essentially recreate or construct the rest of the urethra. And they would do this very early on, a day or two after birth. They would actually go in and surgically reconstruct the rest of that urethra. Right? And I think this here is a good spot to stop. Your head is probably ready to explode. You're probably going to want to go through and watch this a few more times to get a full appreciation of what is happening here really during week 7 through 9 or 10 of embryo development as we look at the development of the gonads. And our next video, get ready for it, we're going to look at the story of the differentiation of sex organs and differentiation, regional differentiation of female and male brain. And I'm going to stress that term, regional differentiation. And so this is where we're going to go next. This is going to get really interesting in the PowerPoint. This is kind of where we're going to look at that variation of sex development and what happens if you've got an overexpression of DAX1 in an XY individual. What happens if you have DAX1 and SRY competing with individuals that are like XXY? What happens when you don't have expected development? Right? What goes on? What does that look like? What does that mean? How does that happen? And so this is going to be where we're going to tie in some of the endocrine system and its influences and impacts with receptors and hormones and all these kind of good things leading into sexual development. And so with that said, I think you know exactly what I'm going to say, and I'll catch you on the flip side.